Hey guys, this video is going to go over the beginning of protein synthesis. And before we really jump into that, this is something that really wasn't mentioned very much in your notes, but is very, very important. So I'm going to touch on it here. I might add a little bit to it, to your slide later. I don't know yet. But hopefully take a second to watch the video. So first thing I really want to talk about is the central dogma of biology. Now, what this is this is how we move from DNA, which is made of nucleotides, and how we transform it into something that can be more readily used in the form of a protein. Now, there are steps to get there, and this is a process that all living things go through. They have to take their DNA, move it to RNA, and then we move it to a protein. Again, this is the central dogma of biology. Now, you'll notice that I have extra arrows out here and a little circle with an arrow here. Now, each of these have a process. Now, I like to include this process on here simply because I feel it's very important. So hopefully you can remember replication. So replication, your DNA has to replicate in order to be passed down to other cells so that they can go through the central dogma. So I like to include replication there. Now, these next two processes, this process right here, moving from DNA to a protein, all of this is called protein synthesis. This is protein synthesis, okay? Protein synthesis can be broken into two processes. Process number one, when we move from DNA to RNA, that process is called transcription. Now, once we have created the RNA, we have transcribed it. So we're taking it from nucleotides to DNA nucleotides, and we're moving them to RNA nucleotides. Now, we need to go a step further. So what we do is we take this RNA and then we make it into a protein, and this process is called translation. Okay. Now, translation. Now, in this video, we're going to focus on transcription, and I'm going to talk about what's important with transcription, and we'll go through an example of a very simple thing of how transcription works. But this process is very, very important because without it, we would not be able to express our traits that we talked about when we talked about heredity and genetics. And so let's hop into it. Let's start crushing through protein synthesis. Let's start with transcription. Again, I said that transcription was moving from DNA to RNA. All we're doing is we're taking that master blueprint that we do not want damaged, and we're going to turn it into something that can be passed on. So we've got our master blueprint, but now we've got to make little blueprints for each of the different components, okay? And that's what this process is doing. So transcription itself will occur in the nucleus, okay? So you need to make sure that when I'm talking about transcription, I'm going to be talking about eukaryotic cells, and we're going to say that this happens in the nucleus, okay? Happens in the nucleus because that's where DNA is found. Now, from there, we have an enzyme, which hopefully you can see here. It is RNA polymerase. Now, that should sound very similar to an enzyme we've already talked about. We talked about DNA polymerase. RNA is very similar. RNA polymerase is very similar, except it makes RNA. And what it does is it does the job of several of those enzymes associated with DNA replication. It does the same as helicase. We don't need primase and DNA polymerase. So it makes and builds this RNA chain. Now, we're going to be focused on mRNA when we're talking about transcription. We'll be focused on the mRNA. Okay? So, focused on mRNA. Because remember, mRNA, that's the blueprint. That's what's going to be used to make these proteins. Okay? So, it takes our DNA, unwinds it, and makes the RNA from it. Okay? So, the enzyme associated with transcription is RNA polymerase. Now, once we have created this pre-mRNA, it's not completely done yet. So it ends up giving us this really, really long strain of mRNA. Okay. Now, we cannot just send it out like this. There are several sections in your DNA that do not code for particular proteins. Okay, so we've got introns and exons. Okay, introns are non-coding regions, while exons are coding regions. The best way to remember that, I know this is hard because it's easy, you can say exit, but exons, they are expressed. Okay, so they make a protein. They're what we need. Okay, introns, left internal. We don't need them. So what happens is we cut these out. We cut those sections out that we don't need. So those introns, goodbye, get out of here. Then we have another enzyme that comes around, and its name is ligase. We've already seen ligase before. Okay, ligase, what it does is once those splices have been done, so we cut out those introns, we cut out those yellow regions, as you see, they get cut out. Ligase comes in and it seals all of these exons together. Okay, so what it leaves is it comes in here, splices all of those regions together, and now we have our mature mRNA transcript that's ready to roll. So what happens is all of this is occurring in the nucleus. We're preparing it, getting it set up. As you can tell, there is a cap and a poly A tail. These are here for extra protection once it leaves the nucleus. And so now once we've got this mature mRNA transcript, we're ready to move into translation. So we got our mature mRNA. All of this process occurred in the nucleus. And now once we have it spliced together, we send it out into the cytoplasm. It leaves the nucleus altogether and it heads to the next section of protein synthesis, which hopefully you know is translation. Now, what we're going to have to do, what you're going to be doing, is you're going to go through, and I'm going to give you 5 questions to work on. And what you're going to have to do is I'm going to give you a strand of DNA, and then you're going to have to give me the mRNA. Okay? So when we're looking here, we have to use our base pairing rules. And this is one of your questions that you have on there. So you're welcome. Now, we've got to remember our base pairing rules. In DNA is A to T, C to G. Now, when we're talking about RNA, remember, there are no thymines in RNA. There is uracil. So when we go and talk about T, T is going to pair with A. Let's do this color. Red. Thymine is going to pair with adenine. Adenine is not going to pair with thymine. It's going to pair with uracil. Keep it consistent. And then cytosine is going to pair with guanine. Okay, so that's our first codon. Remember, our mRNAs are broken into codons. Okay, second codon, this is going to be C. Oh no, it's not going to stay red. It's going to be a pain in the butt. Okay, so we got C, G, C. Boom, boom, boom. Okay, next up, let's put back on red. CCC, CCT is going to be GGA. Okay. Codon 4 is going to be UCC. It'd be fine not being red. Codon 5, CCC. And codon 6 is ACC. Okay. Now, that is how you're going to do this. Now, this is going to be transcription practice. We're going to break transcription and translation up. So what you'll do is after you've taken your notes, you'll start working on the transcription practice. And get done with that, and tomorrow we'll work on translation. All right, y'all have a great day.